All right, let's jump right in. Welcome to this explainer, everyone. I am absolutely thrilled to be your mentor today as we dive into a truly incredible scientific evolution. We're going to trace the journey of Dr. Swee Lay Thein. She's a brilliant physician scientist whose early days as a clinician led her to, well, literally one of the most monumental discoveries in modern hemoglobin research. Now look, this isn't just your standard run-of-the-mill lecture about a single gene. Oh no, this is a story about deep curiosity, relentless persistence, and how a clinician's really keen insight can rewrite the entire future of genomic medicine. We're going to see exactly how a physician transforms into a global genomics pioneer.
So I want you to picture this. Imagine you're doing your rounds, standing right at a patient's bedside. You're looking at two different patients. On paper, their DNA tells you they have the exact same disease-causing genetic mutation for beta-thalassemia. Yet one patient is suffering from incredibly severe symptoms, while the other, they're experiencing unexpectedly mild symptoms, totally free from the exhaustion of constant hospital visits and blood transfusions. How is that even possible? Why do some patients with the exact same genetic error experience completely different clinical realities? This right here, this is the core clinical mystery that sparked a total revolution in blood disorder research.
Here is our roadmap for today's explainer. We're covering 1. The clinical question. 2. From Malaysia to Oxford. 3. Building the statistical foundation. 4. The BCL11A breakthrough. And finally, 5. From discovery to therapy.
Okay, part 1, the clinical question, the bedside mystery. Remember, every great molecular discovery you'll ever make starts right here.
A massive, just a seismic shift in scientific thinking happened at this exact point. When trying to solve this mystery of varying patient severities, Dr. Thein locked onto one massive clue: fetal hemoglobin, or HbF. Clinical data showed that patients who just naturally maintained higher levels of HbF experienced drastically reduced severity in sickle cell disease and beta-thalassemia. It's kind of like a genetic shield. Dr. Thein made a brilliant pivot here. Instead of just staring at the primary globin mutation that causes the disease, she shifted her entire focus to the genetic modifiers, those background genes influencing the whole outcome. She wanted to know, wait, why does HbF stay high in some people but totally drop off in others?
Why do some patients look different? It sounds so simple, right? But this deceptively simple clinical question became the engine driving her entire research career. For those of you studying hematology and human genetics, listen up, because this is the absolute essence of genotype-phenotype correlation. She was so determined to connect the invisible variations in human DNA directly to the real-world clinical outcomes she was seeing on the hospital wards every single day. Moving on to part 2, from Malaysia to Oxford, the making of a clinician scientist. Let's trace how she got here.
So, Dr. Thein was born in Kuantan, Malaysia, and earned her medical degree from the University of Malaya back in 1975. But she had this intense drive to understand the mechanics of blood disorders, which led her over to the UK for some incredibly rigorous postgraduate hematology training. That clinical grounding, absolutely vital. By 1982, she joined the MRC Molecular Hematology Unit in Oxford, and here is where the spark really ignited. Sir David Weatherall at Oxford basically dropped this massive puzzle in her lap. He challenged her to solve that exact clinical anomaly, to figure out why those specific beta-thalassemia patients had such mild symptoms. So how did she actually pull it off? Well, her toolkit evolved right alongside modern technology. Step 1 was pure clinical phenotype collection. She was out there collecting blood samples and, crucially, getting direct feedback from patients and their families about their daily symptoms. That patient engagement was the foundation. Then, as she needed deeper answers, she leveled up. She went to step 2, twin studies, then step 3, linkage analysis. As tech got better, she moved to step 4, genome-wide association studies, and finally step 5, expression profiling. It is just a perfect blueprint of modern scientific progression. All right, part 3, building the statistical foundation and scaling up the data. To prove that fetal hemoglobin levels were truly controlled by genetics, she needed undeniable evidence. So she used classical twin studies. It's basically nature's perfect experiment. By comparing HbF levels between identical twins, who share the exact same DNA, against non-identical twins, the results were just striking. Identical twins had highly similar HbF levels, while non-identical twins were all over the map. This was it. This was the definitive proof the scientific community needed to see that fetal hemoglobin variation wasn't just random or environmental, it was a strongly genetically controlled trait. 5,000. I just want you to let that number sink in for a second. Dr. Thein and her team didn't just look at a dozen patients. They expanded their studies to a massive twin collection of more than 5,000 individuals. The statistical power of a cohort this large is staggering, and it highlights a massive lesson for your own future careers, the absolute importance of scaling up patient data. Without the participation of those 5,000 people, this breakthrough simply wouldn't have had the mathematical weight to be discovered.
Okay, so what do you do with that giant pool of data? Using linkage analysis and quantitative trait locus, or QTL, mapping, Dr. Thein's team basically started hunting for these specific genetic neighborhoods responsible for this trait, and they found them. They successfully identified key regions, specifically on chromosomes 6q and 8q, that were directly tied to fetal hemoglobin production in adults. They were getting incredibly close to the target. Part 4, the BCL11A breakthrough. Zooming into the genome. This is the climax of the story.
By applying GDH draws to her twin cohort, her group found the master switch, a gene called BCL11A. Now, get ready for a crazy plot twist. Before this, BCL11A was known in the medical world primarily as an oncogene, a gene involved in leukemia. Literally, no one expected a known cancer gene to be the master switch for fetal hemoglobin. Actually, scratch that. It completely shocked the field. It was a totally unexpected revelation.
Let's summarize this explosive period really quickly. The GWAS flagged BCL11A. Then, in 2007, Dr. Thein's group definitively proved it was a major repressor of gamma globin. It is literally the molecular brake that switches off our fetal hemoglobin after we're born. By 2008, these findings were published and, super importantly, they were replicated in other populations. Plus, they used expression profiling to link other transcription factors like CMIB to the mechanics of this whole switching process. Guys, this is a masterclass in functional biology confirming a genetic hypothesis.
Finally, Part 5, from discovery to therapy. We are heading right back to the bedside.
Let's connect all the dots here because this is where basic science becomes life-saving medicine. Step 1 was target identification. Discovering BCL11A suppresses HbF. That led perfectly to Step 2, the therapeutic strategy. Think about it. If BCL11A is the brake pedal, what happens if we just take our foot off the brake? That exact logic became Step 3, the blueprint for modern gene editing therapies. By suppressing BCL11A, we can reactivate fetal hemoglobin in adults, dramatically improving the lives of sickle cell and beta-thalassemia patients. Her fundamental discovery literally laid the tracks for next-generation treatments. I really want to leave you with this thought. From a doctor's curiosity to a gene-changing discovery, Dr. Swee Lay Thein's journey is a masterclass in scientific persistence. Her story proves that these breakthroughs, they don't just happen by accident. They start with careful observation, massive amounts of patience, and the courage to follow the data wherever it leads, even if it points to some random oncogene nobody expected. For any of you aspiring medical students or biotechnologists listening, this is your transferable model for success. So, here is my final challenge to you. As you walk those hospital wards, as you comb through those patient charts, keep your eyes wide open. Which clinical anomaly at the patient bedside is going to inspire your next major molecular discovery? The clues are out there. They're just waiting for the next curious mind to stop and ask, wait, why do these patients look different? Keep asking questions, keep chasing the data, and I'll see you in our next explainer.